Welcome to the PubMed Daily Review for May 28, 2026. Today we scanned 458 new open access papers across 82 journals. In this video, we'll cover 10 trending research topics and 10 selected papers for detailed analysis. Trend 1 of 10, Child/Adolescent Mental Health and Development. This trend explores mental health conditions, developmental processes, and interventions impacting children and adolescents. This cluster covers 27 papers. Representative work includes Emotion Regulation and Temperament Characteristics in Children and Adolescents Diagnosed with Migraine and Tension-Type Headache, Interplay of Subjective Symptoms Across the Stages of the Schizophrenia Spectrum, and Global Minds, Caregivers' Perceptions of Early Childhood Development Across Sociocultural Contexts. Trend 2 of 10, Biomedical Modeling and Drug Discovery. Utilizing computational and experimental approaches for biomedical modeling and drug discovery. This cluster covers 29 papers. Representative work includes correction, and a modified HIV model with Beddington-Angelis incidence and cure rate. Trend 3 of 10, Advanced Engineering and Applied Technologies. Developing and applying cutting-edge engineering solutions across diverse fields, from sustainable resource management to advanced electronics and healthcare. This cluster covers 62 papers. Representative work includes additive-free and brine discharge-free solar thermal desalination with simultaneous complete mineral mining from ocean water. Aligners do shed microplastics. Evidence is clear, clinical risk is not, and emerging quality by design optimized HPLC method for the concurrent determination of sulfixime and ornitazole. A multi-criteria green and blue environmental footprinting. Trend 4 of 10, Biomedical Characterization and Epidemiology. Studies detailing biological entities and processes and analyzing health patterns within populations. This cluster covers 80 papers. Representative work includes mapping shared genetic determinants of EGFR and albuminuria with a focus on type 2 diabetes. A large-scale European study, incidence and risk factors of C, trichomonas, N, gonorrhea, and syphilis among a cohort of urban Canadian gay, bisexual, and other men who have sex with men, 2017 to 2023, informing the potential impact of doxycycline prophylaxis, and Zoe cohort, a prospective cohort with comprehensive phenotyping for preventive medicine in France, first 1,000 participants, cohort profile. Trend 5 of 10, Applied AI and Public Health. This trend leverages artificial intelligence and advanced computational methods to address diverse public health challenges and improve healthcare outcomes. This cluster covers 73 papers. Representative work includes primary care for homeless patients, a deep learning approach for solving a fractional order monkeypox transmission model using a harmonic neural network optimized with SGDM, and hybrid topic classification approach integrating TF-IDF, BiGRU, and modified builder optimization algorithm, MBOA. Trend 6 of 10, Cancer Mechanisms, Therapies, and Care. This trend explores cancer's biological mechanisms, therapeutic interventions, and comprehensive patient care, including survivorship. This cluster covers 41 papers. Representative work includes microenvironmental CHRC1 has a pro-tumorigenic role in colorectal cancer, enhanced antimicrobial and anti-cancer properties of penicillin G-adapted lacticacea bacillus rhamnosus GG against colorectal cancer cells, and H. pylori-induced HSA circ 008, 5465 promotes gastric cancer cell proliferation. Migration, an invasion by sponging mere 33b, 3p. Trend 7 of 10, Clinical Study Protocols and Outcomes. This trend features papers outlining clinical study protocols, methodologies, and reported outcomes across diverse medical conditions. This cluster covers 22 papers. Representative work includes telehealth versus in-person caregiver-mediated behavioral treatment for challenging behaviors in children with autism spectrum disorder. Protocol for coach, caregiver outreach for autism coaching at home. Randomized controlled trial, modulate obesity and related metabolic complications for yielding improvements in IBD outcomes. Modify IBD consensus on obesity and cardiometabolic comorbidities in inflammatory bowel disease using systematic reviews and the RAND/UCLA appropriateness method and multi-center prospective cohort study to develop and validate a machine learning-based model for predicting 6-month all-cause mortality in elderly patients with advanced chronic obstructive pulmonary disease in China. Study protocol. Trend 8 of 10, Neuroscience, Imaging, and Neurological Disorders. Investigating brain function, advanced imaging, and therapeutic approaches for neurological disorders across diverse research areas. This cluster covers 48 papers. Representative work includes an ultra-sensitive multi-channel MEG system for the non-invasive single-trial detection of cortical population spikes, predictive metacognition, a neurocomputational framework for self-monitoring in large language models, and neuroimaging outcomes in suspected papilledema. Trend 9 of 10, Health, Nutrition, and Material Evaluation. Evaluating health, nutritional interventions, and material properties across biological and engineered systems. This cluster covers 35 papers. Representative work includes re-evaluating manganese requirements of broiler chickens under phytase supplementation, micro-CT evaluation of aligner fit and thickness in 3D, printed and thermoformed aligners, differences in relation to inner surface offset design and tooth regions, and yoga and the metabolic syndrome, reconsidering an ancient practice in an age of pharmacological abundance. Trend 10 of 10, Environmental Science and Resource Management. This trend explores environmental challenges, resource management, and ecological impacts using scientific methods, often leveraging advanced analytical techniques. This cluster covers 41 papers. Representative work includes a neural network model for managing renewable resources with population growth, assessing the onset of spring water level rise in snowmelt-dominated rivers of northeastern Russia using machine learning, and coastal land uplift and intensified land use influence seagrass carbon and nitrogen sink capacity over millennial timescales. Up next, our first paper out of 10. Phosphometamutase 1 restrains adipose thermogenic programming via inosine signaling by Yafan Yi and Junfeng Han from Department of Endocrinology and Metabolism, Shanghai Diabetes Institute, Shanghai Clinical Center for Diabetes, Shanghai Key Laboratory of Diabetes Mellitus, Shanghai Key Clinical Center for Metabolic Disease, Shanghai Sixth People's Hospital affiliated to Shanghai Jiao Tong University School of Medicine, Shanghai, China. This paper was selected because it identifies a novel and fundamental metabolic regulatory mechanism involving PMM1 and inosine signaling that directly impacts adipose thermogenesis, offering a significant advancement in our understanding of metabolic diseases like obesity. Let's break down its key contributions. Today, we're diving into a fascinating study that explores the role of an enzyme called PMM1 in our fat tissue. Obesity and metabolic diseases are huge problems, and finding new ways to boost energy burning or thermogenesis in fat is a key area of research. This paper asks a fundamental question: how does PMM1 influence our body's ability to burn fat for heat, and what are the broader implications for metabolism? The researchers used a multi-pronged approach. They started by analyzing human fat tissue to see how PMM1 expression correlated with clinical measures. Then, they moved to animal models, specifically mice, and also studied primary fat cells from both mice and humans. They used advanced techniques like RNA sequencing and metabolomics to understand the molecular changes when PMM1 levels were altered, either by reducing or increasing its activity. What did they find? Crucially, PMM1 levels were inversely linked to obesity markers in humans. When they knocked down PMM1 in fat cells, it dramatically boosted thermogenesis and energy expenditure, even protecting mice from diet-induced obesity and insulin resistance. Mechanistically, this happened because PMM1 deficiency led to more inosine, which then activated a specific signaling pathway, ultimately enhancing fat burning. Blocking this pathway reversed the effects, confirming its importance. So what's the big picture here? This research uncovers a brand new pathway where PMM1 acts as a crucial brake on our body's ability to burn fat for heat. By understanding this PMM1 inosine signaling, we now have a promising new target for developing drugs to combat obesity and metabolic diseases. While exciting, more work is needed to see how we can translate these molecular insights into effective treatments for patients. Up next, our second paper out of 10. A multi-society consensus statement on a new common definition and diagnostic criteria for PSVD or NCPF by Virginia Hernandez-Haya and Pierre-Emmanuel Ratu from Barcelona Hepatic Hemodynamic Laboratory, Liver Unit, Hospital Clinic, Clinic Barcelona, FRC Beta BAPS. Fundació de Recerca Clínic Barcelona Institut d'Investigacions Biomèdiques August Pi I Sunior, Barcelona, Spain. ERN Rare Liver, healthcare provider of the European Reference Network on Rare Liver Disorders, CSUR, Centro de Referencia del Sistema Nacional de Salud en Enfermedad Hepática Vascular Compleja en Adultos, Spain. CIBERED, Centro de Investigación Biomedica en Red Enfermedades Hepáticas y Digestivas, Agauer SGR 202101115. Medicine Department, Faculty of Medicine and Health Science, Universitat de Barcelona, Barcelona, Spain. This paper was selected because it represents a critical multi-society consensus statement that unifies fragmented terminology and diagnostic criteria for a complex liver disease, directly addressing a major barrier to research collaboration and consistent patient care in hepatology. Let's break down its key contributions. Historically, conditions like non-cirrhotic portal hypertension have suffered from a confusing array of names and definitions, making it really hard for doctors to diagnose consistently and for researchers to collaborate globally. This lack of standardization meant that patients might receive different diagnoses depending on where they were, and comparing research findings across regions was a major challenge. This paper aimed to tackle this problem head-on by bringing together experts to create a unified approach. To achieve this standardization, a large, international, multi-society initiative was convened. Experts from various liver, pathology, and pediatric hepatology societies across multiple continents participated. They engaged in a structured consensus process, which included specialized working groups and external Delphi validation, to ensure broad agreement and robustness. The goal was to develop a unified diagnostic framework that could be applied worldwide, addressing the inconsistencies that had plagued the field for years. The initiative successfully reached a consensus that the terms portosinusoidal vascular disorder, PSVD, and non-cirrhotic portal fibrosis, NCPF, can be used interchangeably when applying identical diagnostic criteria. They define the diagnosis as fundamentally clinico-pathological, requiring an integrated assessment of various factors. Key principles included the need for a high-quality liver biopsy and mandatory exclusion of cirrhosis. Furthermore, they developed standardized major and minor histologic criteria and an integrated diagnostic scoring system, which was validated using the Delphi method. This consensus statement represents a significant milestone, providing the first internationally endorsed, unified framework for diagnosing PSVD or NCPF. Its global implementation is expected to dramatically reduce diagnostic variability and improve the comparability of patient data and research findings across different regions. Ultimately, this will pave the way for more robust, internationally harmonized clinical and translational research, accelerating our understanding and treatment of these complex conditions. Up next, our third paper out of 10, FAM134B, mediated phagy degrades AP and suppresses Alzheimer's disease pathology by Yuding Zhang and Yixian Shui from Department of Neurology, Department of Urology, Medical Research Institute, Frontier Science Center for Immunology and Metabolism, Zhonan Hospital of Wuhan University, Wuhan, China. This paper was selected because it identifies a novel and fundamental mechanism, FAM134B, mediated phagy, for degrading amyloid precursor protein, AP, which directly addresses a core pathological process in. Let's break down its key contributions. Good morning, everyone. Today, we're diving into the fascinating world of cellular waste disposal, specifically a process called phagy. This pathway is crucial for maintaining cellular health, but its connection to a devastating disease like Alzheimer's has been a mystery. This research aimed to uncover how phagy impacts Alzheimer's pathology and identify key molecular players. To investigate this, researchers employed a multifaceted approach. They used human cell lines to dissect the molecular interactions and then moved to a well-established Alzheimer's mouse model, the 5XFAD mice. They utilized advanced molecular techniques, including AAV gene delivery, to manipulate FAM134B levels and observed its effects on AP clearance, pathology, and even cognitive function in these mice. The results were quite striking. They discovered that FAM134B acts as a specific phagy receptor, directly binding to AP and targeting it for degradation. Importantly, in Alzheimer's, FAM134B's expression was suppressed due to epigenetic changes. When they restored FAM134B in mouse models, it not only cleared AP and reduced amyloid plaques, but also preserved brain health and improved cognitive abilities, highlighting its critical role. This research profoundly impacts our understanding of Alzheimer's disease. It identifies FAM134B downregulation as a critical, early event in the disease process, offering a new perspective beyond just amyloid accumulation. The findings strongly suggest that boosting phagy, perhaps by restoring FAM134B, could be a powerful new way to combat AD. This opens exciting avenues for developing novel therapies that target the root causes of the disease. Up next, our fourth paper out of 10, dual targeting of glutamine metabolism and lysosomal function in eliminating drug-tolerant KRAS mutant cancer cells by Hiroki Furukawa and Shigeki Aoki from Laboratory of Biopharmaceutics, Graduate School of Pharmaceutical Sciences, Chiba University, Chiba, Japan. This paper was selected because it addresses the critical and pressing problem of drug resistance in KRAS mutant cancers by identifying novel metabolic vulnerabilities and proposing a dual-targeting therapeutic strategy, representing a significant. Let's break down its key contributions. KRAS inhibitors are a game-changer in cancer treatment, but their long-term success is often hampered by cancer cells that develop drug tolerance. These persister cells survive initial therapy and lead to relapse, posing a significant challenge. This study aimed to understand how these cells adapt and find new ways to overcome this resistance. Researchers investigated KRAS mutant pancreatic and lung cancer cells in the lab. They used advanced proteomic and metabolomic analyses to understand how these cells adapt to KRAS inhibition. This allowed them to identify key metabolic changes and vulnerabilities in these drug-tolerant cells. The study revealed that KRAS mutant cancer cells enter a drug-tolerant state with significant metabolic changes, including lysosome-linked remodeling. Crucially, they discovered that simultaneously blocking glutamine metabolism and lysosomal function effectively killed these resistant cells. This effect was reversed by alpha-ketoglutarate, suggesting its role in supporting redox balance in these persister cells. This research is significant because it identifies a new vulnerability in drug-tolerant KRAS mutant cancer cells. By targeting both glutamine metabolism and lysosomal function, we might be able to overcome resistance to current KRAS inhibitors. While these findings are exciting, they were primarily in cell models, so the next steps involve validating these strategies in preclinical models and eventually in clinical trials. Up next, our fifth paper out of 10, Transmembrane Domain Switching Controls PINK1 Import and Fate in Mitochondria, by James S. Lorryman and Ian Collinson from School of Biochemistry, University of Bristol, Bristol, UK. This paper presents a fundamental discovery of a novel third pathway for PINK1 import and fate in mitochondria, which significantly advances our understanding of mitochondrial quality control and its implications for neurodegenerative diseases like Parkinson's. Let's break down its key contributions. PINK1 is a really important protein for keeping our mitochondria healthy, and it's strongly linked to Parkinson's disease. We know that when mitochondria are damaged, PINK1 helps clear them out. But what about its role in normal, healthy mitochondria? This paper dives deep into how PINK1 structure dictates its journey inside these vital cellular powerhouses. The researchers used advanced structural modeling and molecular biology techniques to investigate PINK1's behavior. They focused on its transmembrane domain and how it interacts with other proteins within the mitochondria. This approach allowed them to uncover a previously unknown pathway for PINK1 import, providing crucial insights at a molecular level. The study made some exciting discoveries. They found a brand new pathway where PINK1 is actually imported all the way into the mitochondrial matrix. This happens because PINK1's transmembrane domain isn't rigid. It's incredibly flexible, able to switch between an alpha helix and an alpha/beta hybrid structure. This conformational change then determines whether PINK1 goes into the matrix via the ROMO1/TIM17 channel or gets cleaved by PRL and sent for degradation. It's a sophisticated molecular switch controlling PINK1's destiny. This research significantly expands our understanding of PINK1, showing it's not just a damage sensor, but also plays a crucial role in healthy mitochondrial function. The discovery of its TMD's structural plasticity provides a fascinating new molecular mechanism that could be highly relevant for understanding and potentially treating Parkinson's disease. Moving forward, it will be important to confirm these findings in more complex biological systems and explore how this mechanism directly contributes to neurodegenerative processes. Up next, our sixth paper out of 10. Seasonal influenza mRNA vaccine induces stronger innate and comparable or better adaptive responses than licensed inactivated vaccines by Eric Bermudez-Mendez and Karen Lohr from Division of Immunology and Respiratory Medicine, Department of Medicine Solna, Karolinska Institutet and Karolinska University Hospital, Stockholm, Sweden. This paper was selected because it directly addresses a critical global health challenge by demonstrating the superior immunogenicity of mRNA influenza vaccines compared to traditional inactivated vaccines, offering a significant advancement in vaccine technology with broad public health implications. Let's break down its key contributions. Seasonal influenza is a major global health concern, leading to significant illness and mortality each year. While current inactivated vaccines are effective, their production methods are slow, and there's a constant need for more advanced and potent vaccine technologies. This paper explores a promising new approach, an mRNA-based influenza vaccine, comparing its immune responses against the established inactivated vaccines. Researchers conducted a preclinical study using non-human primates to directly compare the immune responses generated by an mRNA influenza vaccine against two approved inactivated vaccines, Vaxagrip and Fluad. They meticulously measured various aspects of immunogenicity, including innate immune activation, the production of HA-specific antibodies, and the development of memory B and T cell responses to understand the underlying mechanisms of protection. The results were quite striking. The mRNA vaccine induced substantially stronger innate immune activation, evidenced by rapid gene upregulation and increased pro-inflammatory cytokines. It also elicited HA-specific antibodies against all four influenza strains, with levels generally comparable to or even exceeding those from the inactivated vaccines. While antibody levels were high, this didn't always translate to greater neutralizing capacity. Importantly, the mRNA vaccine also generated higher frequencies of HA-specific memory B and T cell responses, particularly in the draining lymph nodes, suggesting a robust and lasting immune memory. This study provides compelling preclinical evidence that mRNA influenza vaccines are highly immunogenic, inducing both strong innate and adaptive immune responses. The findings suggest that mRNA technology could offer a significant improvement over current inactivated vaccines, potentially leading to more effective and rapidly deployable influenza vaccines. However, further research is needed to optimize neutralizing capacity and translate these promising preclinical results into human clinical trials, paving the way for a new era in influenza prevention. Up next, our 7th paper out of 10, Personalized Neoadjuvant Treatment Regimen Selection in Locally Advanced Rectal Cancer, based on regimen-specific response modeling by Xiangyu Liu and Xian Wang from School of Life Science and Technology, Xidian University and Engineering Research Center of Molecular and Neuroimaging, Ministry of Education, Xi'an, Shaanxi, China. This paper was selected because it introduces a novel deep learning framework for personalized treatment regimen selection in locally advanced rectal cancer, directly addressing a critical clinical challenge of optimizing patient outcomes and avoiding over- or undertreatment in oncology. Let's break down its key contributions. Welcome. Today, we're diving into a critical area of cancer treatment, locally advanced rectal cancer, or LARC. While neoadjuvant therapy is standard, current treatment selection often isn't personalized, leading to patients potentially receiving too much or too little treatment. This paper tackles that challenge head-on, aiming to bring a more precision-based approach to LARC therapy. The researchers designed an innovative study to address this. They developed a deep learning framework using data from a multi-center cohort of LARC patients. This framework integrated crucial information like pre-treatment MRI scans and clinical variables to predict the likelihood of a pathological complete response for different treatment regimens. They then validated this model in both internal validation and a prospective test cohort. The results were quite promising. The deep learning model showed strong predictive performance, achieving an AUC of 0.827 in the validation cohort and 0.790 in the prospective test cohort. This indicates its ability to accurately predict pathological complete response. Interestingly, the model recommended treatment escalation for over half of the patients, suggesting a potential for more intensive, yet targeted, therapy. It also identified a small group for de-intensification while maintaining high response likelihood. So what's the big picture here? This study presents a significant step towards personalized medicine in LARC, offering a tool that can help clinicians make more informed decisions about neoadjuvant treatment intensity. While the results are exciting, it's important to remember that this is a model. Future research will need to focus on further prospective validation and seamless integration into clinical practice to truly transform patient care. Up next, our 8th paper out of 10. Prediction of antimicrobial minimum inhibitory concentration from bacterial genomes using a scalable and interpretable machine learning approach by Alessandro Girata and William Hope from Department of Clinical Pharmacology and Therapeutics, Antimicrobial Pharmacodynamics and Therapeutics, Institute of Systems, Molecular and Integrative Biology, University of Liverpool, Liverpool, UK. This paper was selected because it introduces a scalable, interpretable, and data-driven machine learning approach for predicting antimicrobial minimum inhibitory concentration directly from bacterial genomes, which represents a significant advancement in combating the global health crisis of. Let's break down its key contributions. Current methods for predicting how well antibiotics work from bacterial DNA are often slow or need a lot of prior information. This makes them hard to use widely. We need a better way to quickly and accurately predict antimicrobial susceptibility directly from a bacteria's genetic code. This research aims to address these limitations by developing a more efficient and interpretable machine learning approach. Our study developed a novel machine learning algorithm to predict antimicrobial susceptibility directly from bacterial genomes. We tested this method using 762 clinical E. coli strains, comparing its performance against existing approaches like those based on known resistance genes or k-mer counts. The primary outcomes focused on prediction accuracy, generalizability to external data, and computational efficiency. Our new model showed impressive accuracy, with 71.6% of predictions falling within one dilution of the actual mic. When tested on new data, it achieved an F1 score of 0.85, outperforming models based on known resistance genes or k-mer counts. Importantly, our method was also much more computationally efficient, using significantly less memory. This study presents a significant step forward in predicting antimicrobial susceptibility using whole genome sequencing. The method is not only accurate and efficient, but also provides valuable insights into resistance mechanisms. This technology holds great promise for improving clinical diagnostics, guiding drug development, and enhancing antimicrobial surveillance efforts globally. Up next, our 9th paper out of 10, Large Language Model versus Multidisciplinary Team. A feasibility study of pancreatic cancer management recommendations by Zhuoran Liu and Kangyi Jiang from People's Hospital of Leshan, number 238 Huyen Street, Shizhong District, Leshan, CN. This paper was selected because it addresses the highly pressing and broadly impactful challenge of integrating artificial intelligence, specifically large language models, into complex clinical decision-making for a difficult-to-treat cancer, demonstrating. Let's break down its key contributions. Managing pancreatic cancer is incredibly challenging, demanding the combined knowledge of many specialists. This complexity often leads to delays or inconsistencies in care. Our research explores whether large language models, or LLMs, could potentially streamline this process by generating initial management recommendations. We wanted to see if these AI-generated suggestions could align with the expert opinions of a multidisciplinary team, potentially offering a new tool to enhance clinical workflow and patient outcomes. Our study was a feasibility evaluation comparing AI-generated recommendations to human expert decisions. We analyzed 125 structured pancreatic cancer cases that had already been reviewed by a multidisciplinary team. For each case, we used a large language model, specifically ChatGPT 5.2, to generate its own initial management recommendations. The primary goal was to assess how well the LLM's broad management categories match those established by the human MDT, providing an initial look into the model's potential in this complex clinical area. We found that the large language model showed remarkable agreement with the multidisciplinary team's initial management categories. Specifically, the LLM matched the MDT in 119 out of 125 cases, resulting in an impressive 95.2% match rate. This finding, with a 95% confidence interval of 89.8% to 98.2%, suggests a strong concordance between the AI and human experts. While promising, it's important to remember this was an exploratory study with a coarse endpoint, and the model was not version-controlled. This study highlights the exciting potential of large language models to support complex clinical decision-making, particularly in areas like pancreatic cancer management. However, it's crucial to acknowledge the limitations, such as the broad nature of the management categories and the specific model version used. Moving forward, these findings strongly advocate for continued supervised exploratory use of LLMs in healthcare. Further research is needed to refine these AI tools and rigorously validate their performance in real-world clinical settings, ensuring they truly enhance patient care. Up next, our 10th paper out of 10, correction, balanced production of ribosome components is required for proper G1/S transition in Saccharomyces cerevisiae by Fernando Gomez Herreros and Mari Cruz Munoz Centeno. Let's break down its key contributions. We're exploring a fundamental question in cell biology. How does the production of ribosomes, the cell's protein factories, impact a crucial step in cell division, the G1/S transition? This transition is a key checkpoint, and understanding its regulation is vital for comprehending cell growth and proliferation. Our focus is on Saccharomyces cerevisiae, a powerful model organism. This study utilized the well-established model organism, Saccharomyces cerevisiae, or budding yeast, to investigate this cellular process. While the specific experimental techniques aren't detailed in the title, we can infer that molecular and genetic approaches were employed to dissect the intricate relationship between ribosome production and cell cycle progression. The core finding revolves around the necessity of balanced ribosome component synthesis. The central finding of this research was quite clear. Maintaining a balanced production of all the various components that make up ribosomes is absolutely critical. Without this delicate balance, the cell's ability to move from the G1 phase into the S phase, a crucial step for DNA replication, was significantly impaired. This highlights a fundamental regulatory mechanism within the cell. This research underscores the profound connection between ribosome biogenesis and cell cycle regulation, a fundamental process in all eukaryotic life. While this specific paper is a correction, it refines our understanding of these intricate cellular controls. Ultimately, insights gained from Saccharomyces cerevisiae often translate to a deeper comprehension of human cellular processes, including those implicated in diseases like cancer. That's all for today's PubMed Daily. If you found this helpful, please like, leave a comment, and subscribe for daily paper summaries. See you tomorrow.